Quantity versus quality. That is the question. Howdy, I'm Doug Tollison, and welcome to the first multimedia edition of the Rimrock Report. In the article that you're reading, when you clicked on this link, we've been having a discussion about what is the most important for grazing animal nutrition. Is it the quantity of forage that the animals are able to get, or is it the quality? And as you read through, you're going to see that we're going to discuss the fact that they both are important, and just because you have half of something that's twice as good as something else, those two things may be mathematically equal, but they're not necessarily biologically equal. And that's what I hope that you will gain from reading this article as it relates to grazing animal nutrition. But again, if you've clicked on this link, it means that you're probably a little more interested in what a plant cell structure might have to do with something that was a high quality forage, such as this green material that we're looking at here, versus something that may be a little lower quality forage, such as this dry dead material. So let's take a look at a plant cell, talk about this in a little more depth, and see if we can't learn something here. So let's talk a little bit about the structure of this plant cell. If we recall back from our freshman biology, and for some of us that's going to be longer than others, we probably had to diagram a cell like this, and we had to remember what those organelles were and draw lines to them and identify them. And probably at that point you thought, you know, if I can just remember these long enough to pass this test, I'm never going to have to know this again. Now here I am dragging it back up for you. But if we look at this cell, we're going to have two main components here that we're going to be interested in. One is going to be the cell wall, and the other is going to be the plant cell contents. Now the wall is just that outer structure, and it's going to be made up primarily of cellulose. Cellulose is the most abundant organic material on the face of the planet, and it is nothing more than long strings, fibers, chains of sugar units, of glucose. But they are bound together with a beta-1,4 linkage. Now why is that significant? That's significant because we, as animals, can't digest those linkages. We can't break them apart because we don't have the enzymes to do it. The ironic thing is we know that neither can the cow or a deer or any other ruminant animal. But because they are ruminants, they have microbes that live in a synergistic relationship with them that do have the enzymes and can break those bonds. Now the other thing we're going to be interested in as you look there is going to be the internal part of that plant cell, and that's where the cell contents are. Now the liquid or the jelly-like substance in the middle is the cytoplasm, and there are going to be compounds floating free in that material that are going to provide nutrients for the animal. But the organelles are going to be the things that we're going to be interested in that are going to provide the bulk of the nutrients out of the cell contents. We might look at the mitochondria, the reddish-brown structures there. We know that's the energy plant, or the source of energy, the engine for the plant cell, just as it would be in an animal cell. That organelle is going to have a membrane around it. It's going to be made of proteins, made of phospholipids, and so that's going to provide some nutrition. Other organelles in there might be the folded structure in the middle would be the Golgi apparatus, and again this is going to have proteins, enzymes in there, also fats. But the thing that the organelle that we're going to be the most interested in is going to be the green structures here, and we know that these are going to be the chloroplasts. We're talking about green plants, plants that undertake photosynthesis and take sunlight, carbon dioxide, nutrition from the soil, and turn that into products that can be used ultimately by other animals. So the chloroplasts, again, are the green structures there, and they have those little stacked disc structures in there. Those are the thylakoids, and this is where photosynthesis is going to take place. Now the key enzyme in that process is rubisco. Now the reason that that's important is because, like cellulose was the most abundant organic compound on the planet, within these compounds, rubisco is going to be the highest, most abundant protein in plants, and thus it's arguably going to be the most abundant protein on the planet. So remember that rubisco in the cell contents is an enzyme which is going to basically provide the bulk of the nitrogen through the microbes to our grazing animals. And so that's why the structure of the cell wall made of cellulose, related to the digestibility, the chloroplast inside the cell, carrying out photosynthesis with this enzyme rubisco, providing the bulk of the nitrogen and thus ultimately the protein. Okay, let's talk a little bit now about how that cell structure that we just discussed is going to be important and how it relates to actual physical structure of plants and selection of diets by animals. We know that if animals have their choice, if we've provided an adequate quantity of forage for them to be able to do so, they're going to be selective in their diets, and they're going to select a better diet than what would be just the forage on offer if we just go out and take clip samples and do chemistry on that. And we know that the animals, if they have their choice, they're going to choose more live material as opposed to dead material in a plant. They're going to choose more new growth as opposed to old growth, and they're going to prefer leaf over stem. So the term new live leaf is going to be that most highly selected portion of the plant if an animal has the ability to make its choice. So how does that relate then to plant cell structure, and why is that important to this whole discussion about quality and quantity of animal diets? Well, if we think of those plant cells as being a lunch, okay, and if we want to think of that as being either a sack lunch or a box lunch, I think we'll be able to make the point. Now, when I fix a lunch, I'm going to do this pretty rapidly. If I'm going out to work, I'm going to grab a sack, I'm going to take 2 pieces of bread, put some bologna between it, grab a bag of chips, put it in there. That's going to be about all I'm going to do. But when my grandmother used to make me a lunch, she was a lot more meticulous about it. And she would either put it in a sack and then put that in another sack, or sometimes she would put it in a box, a cardboard box, not a styrofoam box like this. But she might put that in there, and she would put napkins in it, and she would put cookies and goodies and all sorts of things in there, and then she would probably tape it up so that it wouldn't spill out during the day. Well, if we want to think of these two as being symbolic of plant cells, something with a cell wall around it and with contents on the inside, a sac lunch, which if I'm a bacteria in the rumen and this is a plant cell, is something that I'm going to be able to get into fairly easily and get the goodies out of. Okay, not a lot of cellulose, thin cell wall, pretty easy to break, tear apart if I'm a bacteria. That is going to represent the type of material that we would find in green, lush, or new live leaf. Okay, new live leaf is going to be represented by a sac lunch. Now, on the other hand, if I'm a bacteria and the plant cells that I've been presented in the rumen are from old, dry, dead, or stemmy material, I'm going to be looking at a whole lot of box lunches. Now, box lunches are going to be thicker, more cell wall, more cellulose, and I can eventually get into them, but it's going to take a little bit more work. I'm going to have to tear through some different things. It's going to take me some time, but eventually I will get in there and also get at the goodies, the cell contents. Now, on a weight basis, if this box lunch weighed the same as the sack lunch, there's going to be more of that weight in cellulose, in harder to digest material, as compared to the more readily available cell contents. So, as we look out across a pasture and we're trying to determine what the nutritional ecology of the animals that are going to be residing in that pasture are likely to be, remember that that is going to be a combination of sack lunches versus box lunches, and so the plant cell structure, as it relates to the overall plant structure and the diet selected by the animals, that's going to be a very important determinant in this quality versus quantity debate. So remember, sack lunch, new live leaf. Box lunch, old dead stem.